This lecture is about cellular reproduction. So, in order to have an organism grow or reproduce, cells have to make new cells. And they need to pass on the genes from one cell to the next. So before a parent cell divides, it has to duplicate its chromosomes, which contain DNA. And a very simple way of division has two daughter cells resulting that are both genetically identical to one another. And simple single-celled organisms reproduce with this sort of simple cell division, and we call this asexual reproduction. But we're interested in learning about two different forms of reproduction of cells. The first is mitosis, the second is called meiosis. We'll be getting into those in quite a bit more detail, but a simple way to remember the difference between them is mitosis happens in your toes. It's the division that results in new cells that are the same as each other, and meiosis happens in your eggs or your sperm cells. That's how you're making new reproductive cells. Well, let's get started with the idea of the genome. The genome is the complete set of an organism's genes. These are sequences of DNA that code for specific proteins. And the DNA is located mainly in the cell's nucleus on short fragments of DNA referred to as chromosomes. A chromosome is actually made out of a combination of DNA and protein molecules, and most chromosomes are not visible in the cell until the cell is about to divide. The DNA gets packaged around these proteins called histones in a really elaborate system of folding, and it folds so much so that you can actually see a chromosome under a light microscope. So before a cell divides, it has to make sure that it has one copy of the genome to put into each new daughter cell. So the DNA has to undergo DNA synthesis, which is sometimes called chromatid duplication. And what results are two sister chromatids. These are two fragments of DNA that are identical to one another. And during cellular division, the chromatids are distributed one to each new daughter cell. Each cell undergoes a sequence of events called the cell cycle. It has two phases. The first phase is called interphase, and the second is called the mitotic phase. During interphase, a cell prepares for DNA synthesis. So it makes all of the new adenine, thymine, cytosine, guanine. It gets prepared for synthesis. It makes the new DNA. So you go from having one copy of the genome to two copies of the genome. Then the cell prepares itself for the mitotic phase. The mitotic phase, this is the M phase, is where we have mitosis. And during mitosis, the chromosomes separate and are allocated into new cells, and the process of cytokinesis breaks the new cells into two, and each of those new daughter cells enters interphase. So, mitosis itself consists of four distinct phases: prophase, metaphase, anaphase, and telophase. Interphase, which is shown on this panel here, is actually not strictly a part of mitosis, because interphase can happen before mitosis, or it can happen before meiosis. So interphase is just that time between cell division periods. So what are the four phases of mitosis about? Well, I want you to realize that while we define them into four distinct phases, this is a continuous process. So during prophase, chromosomes form. So where you would initially have a diffuse colored nucleus, you'll start to see little tiny sausage-like structures, and those are the chromatids forming. You also have the formation of an internal structure called the mitotic spindle. This structure is like a network of railroad lines that help the chromosomes to separate appropriately. During metaphase, the chromosomes line up along the middle of the spindle. That's what meta means. It means middle. The chromosomes hook themselves up onto the mitotic spindle, and then the two sister chromatids get popped apart. During anaphase, those two sister chromatids, now called daughter chromosomes, move towards opposite sides of the cell. During telophase and cytokinesis, the new daughter cells form and the nuclear membrane starts to form around the two new nuclei. Cytokinesis is a little bit different in plant cells and in animal cells. In animal cells, you have the formation of something called a cleavage furrow. You don't actually get that in plant cells. So now let's briefly talk about the division of your genome into smaller fragments. The human genome, if you were to have all of the DNA lined up end to end, would be approximately 6 feet long. So imagine now you've got two strands that are 6 feet long, crammed into the size of a single cell, which you can barely see, or not see at all. That would be very difficult to move the two strands to opposite sides of a new cell. It would be like trying to untangle two large balls of wool that had been tangled up by a cat. So nature's way of having successful cell division is to break up the genome into small chromosome fragments. A karyotype is the description of how the genome is broken up into small fragments, and this actually varies dramatically between species. In humans, the genome is broken up into 23 fragments, and you get one copy of your genome from your mother and one copy of your genome from your father. And as a result, in your cells, you have 46 fragments, 23 from mum, 23 from dad. A homologous chromosome is a chromosome that is part of a matching pair. So in this image here, you'll see chromosome labeled number 1, and there's 2 of them. One of them came from the mother and one came from the father, and so on and so forth. This individual happens to be male. They've got one X chromosome and one Y chromosome. So of the 23 fragments, 22 of them are what we would call autosomal chromosomes. Those are the chromosomes that code for most of the body functions. The last set of chromosomes are called the sex chromosomes, and you can have an XX, which codes for a female, or an XY, which codes for a male. So it's very important that during cell division, especially the cell division that results in sperm and eggs, that you have appropriate allocation of homologous chromosomes. You need one copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 3, etc. in each new sperm cell or egg cell in order to have successful reproduction. So gametes are very important in the life cycle of sexual organisms like humans. The egg cell and the sperm cell contain only one copy of the genome. They're called haploid. So they have 23 chromosomes within them. Upon fertilization, when the sperm and the egg fuse to form a diploid zygote, you have 2 full copies of the genome. The diploid zygote then grows and divides through mitosis and subsequent cellular differentiation to create a multicellular diploid adult. those diploid adults in their ovaries and testes will have cells that undergo meiosis to then create the new sperm and egg cells. So this diagram here shows the process where a homologous pair of chromosomes during interphase will duplicate. So you actually end up with, you start with 2N, 2 copies of the genome. You end up at the end of interphase, before you start meiosis, with 4 copies of the genome. And in order to create egg or sperm cells, you have to go through 2 rounds of cellular division, meiosis 1 and meiosis 2. So during meiosis 1, the homologous chromosomes separate, and during meiosis 2, the sister chromatids separate. So meiosis has got a couple of extra things that are different than mitosis. So let's pay attention to those. During prophase of meiosis 1, you have the formation of something called tetrads. A tetrad is where 2 homologous chromosomes that have duplicated and each consist of 2 sister chromatids align. So what you have are 4 copies of the same chromosome hanging out together. Tetra means 4. And when they hang out together, they have what are called crossing over sites. So where 2 pieces of DNA cross one another, they can actually have exchange of genetic material. So part of a maternal chromatid can be exchanged with part of a paternal chromatid. And what this does is it reshuffles the genes. So you can have your grandmother's eyes and your grandfather's chin. During metaphase, you have the chromosomes lining up along the middle. Now, how they align at the middle can vary. You could have both of the paternal chromosomes being at the top, so all paternal chromosomes going towards one side of the cell, or you could have all the maternal chromosomes going to the other side of the cell, or you could have some of the paternal and some of the maternal chromosomes. This is called independent assortment. Between crossing over and the process of independent assortment, you can create a huge amount of variability in the genetic makeup of the gamete cells that result. So where in mitosis the mother cell and the daughter cells were identical, in meiosis the daughter cells are different from each other and they're different from the original cell. So if we've continued through anaphase 1, the homologous chromosomes break apart. Now the homologous chromosomes have non-identical sister chromatids because of that crossing over. So notice here you've got a little bit of blue and you've got a little bit of red. And you form 2 haploid cells. The chromosomes are still doubled at this point. In meiosis 2, the non-identical sister chromatids align along the middle point, the metaphase 2 plate, and then they break apart, moving to separate parts of the cell, forming the haploid daughter cells. So to compare mitosis and meiosis, mitosis you have 2 daughter cells that are identical to the parent cell, whereas in meiosis you have 4 haploid daughter cells that are non-identical and they're not the same as the original parent cell. Now, independent assortment is very important because it creates variability in the gamete combinations. So does crossing over. Crossing over results in recombinant chromosomes where you have combinations of maternal and paternal genes. Now, what about if meiosis goes wrong? What about if the homologous sister chromatids don't move apart correctly, or the non-identical sister chromatids don't move apart correctly? Well, this process is called nondisjunction. And nondisjunction results in gametes that have the wrong number of chromosomes. And this can cause problems for a developing fetus. because the zygote that results from fertilization of an egg that has the wrong number of chromosomes will also have the wrong number of chromosomes. An example of a human disorder that results from nondisjunction is called trisomy 21 or Down syndrome. This is where an individual has inherited an extra chromosome. There are several other instances of nondisjunction resulting in human disorders. One of them is trisomy 18 and another is trisomy 13. Those two disorders, unlike Down syndrome, which can result in an individual who, while they may suffer some disabilities, they're actually still alive. Trisomy 13 and trisomy 18 are not compatible with life. A fetus may die in utero, or at most might have 3 or 5 days of survival after birth.